ever wondered why identical twins are always different even though they have the same DNA and should perfectly resemble each other? Until very recently, it was thought that DNA dictated every single trait of ours, from the color of our eyes to how good we are at maths in school. But scientists noticed inconsistencies in the laws of genetics that could not really be explained, and they realized that there is something else at play, controlling the way that genes express. Epigenetics literally means above genes, and it studies how the environment influences the way that cells in our bodies read genes through various mechanisms that don't change the DNA sequence itself. DNA is practically a blueprint for different types of proteins that a cell can create, as well as other functional molecules. In order for a protein to be synthesized, DNA first gets transformed into a complementary strand of messenger RNA. This mRNA exits the nucleus and gives ribosomes instructions to assemble amino acids into a protein. Epigenetic mechanisms, or the gene police as I like to call them, intervene anytime during this process. The addition of chemical tags to the DNA, such as methyl groups to cytosine in certain regions of genes, can silence them. The tails of histone proteins that DNA is wrapped around can be methylated, causing DNA to wrap tighter and become inaccessible, whereas adding an acetyl group will cause the DNA to loosen. Non-coding RNAs do not code for any protein but have a vast range of action. Some of these will bind to mRNA and prevent it from being read or splice it into pieces, and they can even stop ribosomes from assembling and reading the mRNA. How does a cell know when to silence or activate a gene? It all comes down to whether the concentrations of certain substances like hormones, enzymes, proteins, and others are within certain limits. Before we are born, most of the tags from our parents are wiped away, but some get passed down through multiple generations. Everyone has little epigenetic ghosts from their predecessors that can have a big impact on their lives. Fortunately, our epigenome is highly versatile. In babies, most genes are methylated, but as we grow up and our bodies interact with our constantly changing environment, these genes demethylate. And when we are old, we reach the lowest levels of methylation. But which genes get turned on or off depends almost entirely on our lifestyle. Without our epigenomes, we would probably look like huge blobs that can't do much because our stem cells wouldn't be able to specialize in order to perform certain functions, like muscle cells that contract or neurons which conduct electrical impulses. Thank you. When we learn new information, our neurons undergo intense epigenetic changes that allow them to form new and efficient pathways. Even cancer cells have some bad genes turned on and good ones off, enabling them to divide chaotically. It's not just genes that make us. DNA does not dictate our fate from the moment we are born. It simply tells us what we can be, but it is our responsibility to decide what we become.